All right, now we need to talk about cell-mediated immunity. Adjust my pencil a little bit, sorry. All right, so cell-mediated immunity, if you remember, we did the overview with the cells, the target for cell-mediated immunity is going to be intracellular pathogens. All right, so intracellular pathogens, we're talking viruses, we're talking cancers, things that are going on inside of a cell, not floating around like a bacteria in the extracellular fluid. So we have the naive T cell. And the naive T cell, of course, has his T cell receptors. All good. Okay. So the naive T cell, as I said, the T cells look for intracellular pathogens, so they don't see their antigen floating around. They don't respond to the antigen on their own. Okay? Naive T cells must be presented its antigen. So naive T cells must be presented its antigen. They don't see it. They don't, if it's floating around, they're blind, they don't see that, okay? So it has to be shown the antigen with an antigen presenter, so an antigen presenting cell. So the antigen presenting cell then will show the antigen on its MHC molecules, okay? So how does this occur? Well, we have two different types of cells to deal with, the helper T cells and the cytotoxic T cells. So helper T cells are shown their antigen through an antigen presenting cell, usually a dendritic cell. And they present to a naive T cell that is a CD4 cell. They present on an MHC2 platform. So if this is a CD4 cell, then it will bind to this antigen presenter with its MHC2 platform. Okay? The antigen presenting cell will activate the CD4 cell. Okay? So once it binds, so let me erase this and show it binding. Okay, so here's my little T cell receptor, and then here's my MHC2. So it will bind. Okay, so they bind together. In T cells, the binding of these T cells is not enough. Okay, the antigen presenting cell or dendritic cell will supply something called a co-stimulatory molecule. And the co-stimulatory molecule must be there. And co-stimulatory molecules, they're going to be more co-stimulatory molecules for helper T cells than they're going to be for cytotoxic T cells. Because we need more helper T cells, because helper T cells activate other cells, including cytotoxic T cells, and of course B cells. So once the co-stimulatory molecule is there, then the naive T cell becomes activated. So activated. Okay, and it becomes activated into a helper T cell. Once it's a helper T cell, it goes through the same thing as the B cells. It goes through clonal expansion or whatever. Yeah, clonal expansion. They divide, and it makes more helper T cells. Okay. The cytotoxic T cells also have to be shown their antigen by an antigen presenting cell, and it's going to be a dendritic cell. Dendritic cells are unique because dendritic cells have both MHC1 and MHC2. Okay, so the cytotoxic T cell, so let's look at the cytotoxic T cell, also, it's a CD8 cell. So if that naive T cell is a CD8 cell, then it will, so naive cell, and we'll call it a CD8 cell. Okay, so it'll have its little receptors, right? Little T cell receptors all ready to go. Okay, so it will bind to an antigen presenter. So the APC. Well, the APC is going to present the antigen on MHC1. Okay, MHC1, remember, MHC1 is on all body cells, and MHC1 is where a body cell is going to post its virus or whatever on its plasma membrane, and that's how that cytotoxic T cell is going to find it and latch onto it, because it knows to latch onto MHC1. Okay. Again, we have to have co-stimulatory molecules in order to be activated. So we have the co-stimulatory molecule. And activation can occur. The co-stimulatory molecules on antigen presenting cells are finite, so there may not be enough of them. If that's the case, then a helper T cell can come over and activate the naive, or activate the CD8 cell. So if we have a CD8 cell here, let's do so that I have enough room. So here's the APC. All right, so it is trying to activate CD8. And we'll put CD8 down here. And it looks like that. Do that, whatever. Okay, so it's bound to that, and it's trying to activate that, but there's no co-stimulatory molecule. So let's say that there isn't one. There's no co-stimulatory molecule here to match. All right. a helper T cell a little bit of a different color here. So a helper T cell, so a helper T can come over and it will bind to the same thing, and it will bind to the APC's MHC2. So they're bound together, and then the helper T cell then can send molecules over to CD8, so interleukins. So those are just cytokines, and we can get that cytotoxic T cell activated. Okay, so the T cells are a little bit different. The CD8 cell, once it's activated, it will go through clonal expansion. And when they go through clonal expansion, whether they're helper T cells or cytotoxic T cells, they make more of themselves, and then they also make memory cells. And the ones that they make of themselves are going to go looking for body cells that have MHC1 posting their antigen. And then they're going to latch on to that body cell and cause it to go through apoptosis. Okay, so here's the thing too. So let's say we don't have co-stimulatory molecules and there's no helper T cell, because co-stimulatory molecules really are only there when the body has alerted that there actually is an infection. Okay, so if there's no for sure infection going on, then there's not going to be co-stimulatory molecules that are going to be generated. So if that's the case, and there is a CD8 cell that is latched onto an APC, and it doesn't have co-stimulatory molecules, and there's no helper T cell, then the CD8 cell does not get activated. And if it does not get activated in this instance, it becomes anergic. So something called anergy. And once it's anergic, it is always anergic. Once it's anergic, it will never, ever, ever be able to be activated into an active T cell at that point. All right, so that right there is cell-mediated immunity. And next, we're going to go through hypersensitivity reactions.